Hello everyone, I am Dr. Omkar Sangita Dilprao Sonone, and today we are beginning the FMG June 2022 preparations. So today onwards we'll be having regular sessions on YouTube and various other platforms. So first of all, before beginning, I would like to give you an insight of how this journey of 4 months is going to be. So please remember guys, just like last time also, AFMG will be your partner in your preparations this time too. Okay, these 4 months will be having different types of sessions. First of all, if I talk about our regular batch, which has already been started, and the last batch is about to start very soon. So if you want to join the regular batch, please do contact AFMG via its various social media handles or the number you can find in the description below. Okay, so please contact AFMG from there itself. We will be having offline face-to-face classes in Delhi, Chennai and Trivandrum, and also we are having our online classes too. So if you wish to join any of the batches, please do contact. Second thing, the second initiative that we are about to take is the YouTube that we have been continuing for the last session, and we will continue for this session too. So on YouTube now I'll be coming up with some videos from different subjects of a very important or high yield topics. Those have been recently asked, or there's a high probability that these topics can be asked. Okay, so such topics we'll be discussing, not in great details, because the primary source of your revision should be your notes, but this would definitely help you clear your concepts about that particular topic so that you do not repeat any mistakes in these particular topics at least. Okay, so with this we are also beginning a new test and discussion series which will be starting from 23rd of January itself. For that also you need to register and enroll yourself with the numbers which are given below. The third thing I want to highlight is you need to join the AFMG Telegram group, that is by the name AFMG for FMGE/next. It is for your benefit because over there you can get ample amount of MCQs regularly on day-to-day basis to solve and to practice. Apart from that, over there only in the same group you will be receiving all the updates regarding each and every session, may it be on your YouTube or offline or online session of AFMG. So every update you will be receiving over there, and also you will receive all the updates and guidance for your FMG June 2022 preparation. Okay, so if any issues you can just comment down in the chat box itself and we'll be more than happy to answer your doubts regarding those. Okay, so with that I will start with the most basic subject, and that is anatomy, which is most important to understand the clinical subjects like orthopedics and surgery. So anatomy, today we'll be discussing embryology, which is an important topic and it is a bit complex topic. So I have narrowed it down to you. That does not mean your source of notes should change. The source will remain your notes. This session would help you make your concepts more clear and will narrow down your notes. At least you need to know these topics which have been discussed in these 2 sessions. Yes, embryology will be discussed in 2 sessions, the one which I am recording right now and the other one I'll come up with soon. Okay, so these are the questions we'll be discussing which have been asked earlier, or there's a high probability that these questions from in and around these topics can come in the examination. Okay, so without any further delay, let's begin our session for today, that is embryology. So if I start with embryology, embryology means study of the embryo, right? So now what do we mean by embryo? So please remember, when a baby is growing inside the womb in the intrauterine life, it is called the embryo, but it does not mean that it is called embryo throughout the intrauterine life, right? So please remember, after fertilization zygote formation occurs. So from fertilization till 2 weeks, it is known as the pre-embryonic period. So please remember, after fertilization till 2 weeks, this is known as your pre-embryonic period. Then from 2 weeks onwards till 8 weeks of intrauterine life. Okay, from 2 weeks till 8 weeks of your intrauterine life, the baby would be called an embryo, and this is known as the embryonic period. Okay, from 2 to 8 weeks, this is known as your embryonic period. Why it is important to remember? Because this was the question which has been asked. Embryo means from 2 to 8 weeks of intrauterine life. Next, if I talk about from 8 week onward, after 8 weeks, or we can say from 9 week onwards. So from 9 weeks of intrauterine life till birth, now the baby would be called as a fetus. Now the baby is called as a fetus, and therefore this period is known as a fetal period, right? So I hope that you do not make any mistakes. 2 to 8 weeks is known as the embryo. After that, fetus. Then if I talk about embryology proper now. So first of all, I'll be starting with the primordial germ cells. Okay, so I hope that all of you know that these primordial germ cells usually they are pluripotent cells. What do we mean by pluripotent cells? That means they can differentiate in all the 3 germ layers. I hope you know all the 3 germ layers, that is your endoderm, your mesoderm and your ectoderm, right? These are the 3 germ layers, and these pluripotent cells, they have the ability to differentiate in each of these. Okay, and such ability is seen with primordial germ cells, therefore they are also known as pluripotent cells. Now usually these primordial germ cells, they travel or migrate to the gonads, that is the ovaries or the testis, till 5th week of intrauterine life. Okay, till 5th week they travel or migrate to their gonads. But what if these primordial germ cells also migrate abnormally to other regions? So if there is abnormal migration of primordial germ cells in the head and neck region, that would lead to a formation of a craniopharyngeal teratoma. Okay, this would lead to a formation of a craniopharyngeal teratoma. Whereas if these primordial germ cells, they migrate to the sacrococcygeal region, that would lead to formation of a sacrococcygeal teratoma. That would lead to formation of a sacrococcygeal teratoma, for which the image has been given below. And this was a question which has been asked. So here you can see at the sacrococcygeal region, there is a tumor or a mass arising. This is known as a sacrococcygeal teratoma. Okay, so please remember, what do we mean by a teratoma? Then oma means it is a tumor. Right, so teratoma is such kind of a tumor which contains all the 3 germ layer derivatives. That means it usually arises, the teratoma arises from the primordial germ cells. Okay, so teratoma is any tumor which arises from the primordial germ cells, and these primordial germ cells has the ability to differentiate into all the 3 germ layers. Therefore, in any kind of teratoma, we can find hair, nail, bone, mucosa, skin, teeth. Okay, all of these derivatives can be found of all the 3 germ layers. Okay, such type of tumors are known as teratoma. So this image had come, if it arises from the sacrococcygeal region, sacrococcygeal teratoma. If it arises from the craniopharyngeal region, craniopharyngeal teratoma. Next, now I will talk about a most important topic, and that is gametogenesis. Okay, gametes are, in females it is the ova or the egg, whereas in males it is the sperms, also known as the spermatozoa. I hope you are clear about this. Now gametogenesis would be oogenesis in females and spermatogenesis in males, right? So if I talk about females first, okay, that is oogenesis. As we are quite aware that these primordial germ cells in the gonads, they will further differentiate into oogonium first. Right, after oogonium is formed, that will further differentiate into the primary oocyte, that will further differentiate into the secondary oocyte, and finally that would lead to formation of a ova or the egg, female egg. Right, so now what happens usually, these primordial germ cells are the only cells in the body which has the ability to undergo meiosis. I hope you are quite aware of cell division. So there are 2 types of cell division, mitosis and meiosis. All the other body cells undergo mitosis, whereas the primordial germ cells undergo meiosis. Okay, where the chromosome number goes half, that is haploid cells are seen, and gametes are haploid cells, right? So usually the primary oocyte and the secondary oocyte undergo meiotic divisions. Okay, so first meiotic division and second meiotic division. But what happens is the primary oocyte is arrested in the first meiotic division. In which phase this question has been asked. So please remember, primary oocyte is arrested in the prophase of the first meiotic division. Okay, primary oocyte is arrested in the prophase of the first meiotic division. Whereas the secondary oocyte, it is arrested in the metaphase of the second meiotic division. Secondary oocyte is arrested in the metaphase of the second meiotic division. So please remember, after first meiotic division, primary oocyte is formed and first polar body is formed. Okay, and it is arrested in the prophase itself. It completes the meiosis 1 after puberty. then it undergoes meiosis 2 and a secondary oocyte is formed with second polar body, and it completes this second meiotic division after fertilization. Okay, so please remember these two are the most important points that you need to remember over here. Okay, and after the second meiotic division, then an ova is formed. Now if I talk about this, guys, this was about your oogenesis. Now if I talk about spermatogenesis, the sequence has been asked in the examination. Okay, so what is the sequence of spermatogenesis? First of all, the basic unit would be the primordial germ cells, from which the spermatogonium would develop, from which the spermatocyte would develop, again a primary spermatocyte and a secondary spermatocyte, which will undergo meiotic divisions. Then spermatids would develop, and finally a spermatozoa or a sperm would develop. Now please remember what are the major two differences in oogenesis and spermatogenesis. Okay, in oogenesis in females there are two polar bodies formed. Okay, so please remember in oogenesis there are two polar bodies formed, which are not seen in cases of spermatogenesis. Whereas the second difference, from one oocyte usually only one single ova develops. Okay, from one oocyte single ova develops, but from one spermatocyte, from a single spermatocyte usually 4 spermatids and therefore 4 sperms develop. Okay, so these are the major two differences that you need to remember. Moving further, now talking about some important one-liners of these processes. Now if I talk about the duration of spermatogenesis, so please remember usually the duration of spermatogenesis is 74 days. Now this is a question from 12th standard itself. So duration of spermatogenesis is 74 days. Okay, this is for the formation of sperms, which is known as spermatogenesis. And this formation of sperms occurs where? The site of spermatogenesis is what? So please remember the site of spermatogenesis is the seminiferous tubules of testis. Okay, the site of spermatogenesis is the seminiferous tubules in the testis. Right, now after the sperms are formed, they are required, okay, it is compulsory for them to stay for 12 days for the purpose of maturation and motility. So they stay in the male genital tract for another 12 days for the purpose of maturation and motility, and this undergoes in the epididymis. So please remember this process of sperm maturation and motility takes place in the epididymis. So please remember sperms stay for another 12 days in the epididymis, and therefore epididymis is also known as the storehouse of sperms. Right, now if they ask you about this, questions have been asked earlier, what is the lifespan of a sperm and what is the lifespan of an ova? So please remember the lifespan of a sperm is considered to be 96 hours. Okay, lifespan of a sperm is considered to be 96 hours, whereas that of an ova is considered to be 24 hours. That of an ova is only 24 hours. And this is where the fertile period comes into action. So please remember the fertile period that we say, or the unsafe period of the menstrual cycle, is when the ova can fertilize. Okay, and it is considered from the 11 to 16 days of menstrual cycle. This is an important question from OBG. So please remember fertile or unsafe period is from 11 to 16 days of menstrual cycle, because we consider ovulation usually occurs on 14th day. Ovulation usually occurs on the 14th day. Now if I move further, I will talk about fertilization now. Okay, so please remember, what do we mean by fertilization? Fertilization means fusion of the male pronucleus with the female pronucleus. This fusion is known as fertilization, right? And this actually occurs with various reactions. First is the acrosomal reaction, then a cortical reaction, and then a zona reaction, right? So important is, please remember only one sperm can fertilize a single ova. Okay, after that, to prevent polyspermy, usually the zona reaction occurs. The zona pellucida does not let the other sperms to come inside. Okay, so please remember only one sperm can fertilize a single ova. So this is known as the process of fertilization. So what is the site of fertilization? Question has been asked. So please remember the site of fertilization is the ampulla. So please remember the ampulla of the fallopian tube. Okay, so please remember the ampulla of the fallopian tube is the site for fertilization. Once the ova is released from the ovary into the fallopian tube, it is taken up by the fimbria. Then it goes to the widest part of the fallopian tube known as the ampulla, where the fertilization would occur. Right? Now there was a question in recent December 2021 session that what kind of epithelium is seen in the fallopian tube. So this fallopian tube, also known as oviduct, also known as uterine tube, in this the epithelium which is present is a seriated columnar epithelium. This is a question which has been asked twice. So please remember, the epithelium present in the fallopian tube is a seriated columnar epithelium. Why do they require cilia? For propulsion of the ova. It moves the ova forward. Okay. And a question which has been asked previously, that which specialized cells are present in the fallopian tube. So the specialized cells which are present in the fallopian tube, these are known as the PEG cells. Okay, these are known as the PEG cells, and these PEG cells usually have a function of nourishment of the ova, right? Important. Next, now after the fertilization occurs usually, then there is formation. So please remember, after the fusion of fertilization, there is formation of a zygote. Okay, the structure which is formed is known as a zygote. Now this zygote will undergo multiple cell divisions known as the cleavage. Okay, it will form a 2-cell stage, then a 4-cell stage, then a 8-cell stage, and moves on. So usually this 2-cell stage is usually formed on the second day after fertilization. It is formed on the second day after fertilization. Then it moves ahead, and then finally a 16-cell stage is formed. A 16-cell stage is formed which is known as a morula. So this was a question which has been asked. Morula is a 16-celled stage of a zygote. Okay, please remember. And morula is formed when? So this is also important. So please remember, morula is formed on the 3rd day after fertilization. Morula is formed on the 3rd day after fertilization. Important, right? Now, after morula formation on the 3rd day, this morula will enter the uterine cavity. Okay, and when does the morula enters uterine cavity? So please remember, morula enters the uterine cavity on the 4th day after fertilization. Then next, after it enters the uterine cavity, there is formation of a blastocyst, and blastocyst is formed on the 5th day. Some books say it 4.5, so it's fine. 4.5 to 5th day after fertilization, there is formation of a blastocyst. So what do we mean by a blastocyst? So please remember, the cell mass which was there, now it differentiates. Okay, it differentiates into this inner cell mass which is known as the embryo blast. Okay, the inner cell mass known as the embryo blast, and this outer cell mass known as the trophoblast. Okay, so it differentiates into 2 cell masses, and in between a cavity is formed known as a blastocele. Okay, cavity is formed known as blastocele, therefore this is known as a blastocyst. And after this blastocyst formation, then the final process occurs of implantation on the endometrial wall. Okay, and this implantation of blastocyst usually occurs on the 6th day. It starts on the 6th day after fertilization. Now coming back to our one-liners, now you can just answer those very easily now. Morula is formed on, so please remember, morula is formed on 3rd day after fertilization. Next, zygote enters the uterine cavity on, so please remember, zygote enters the uterine cavity on the 4th day after fertilization. Next, please tell me, blastocyst is formed on, so blastocyst is formed on the 5th day after fertilization, right? We discussed. Now if I talk about implantation, so please remember, implantation starts at, implantation starts at 6th day after fertilization, but it completes by 9th day after fertilization. So please remember, the implantation window is from 6th to 9th day. It starts on 6th day and completes by 9th day of fertilization. Next, now as we discussed, the inner cell mass was known as the embryo blast, and this embryo blast further develops into the embryo proper. This embryo blast further develops into the embryo proper. Whereas the outer cell mass, it is known as the trophoblast, and this trophoblast further leads to development of the placenta. I hope all of you are quite aware that placenta has 2 types of cells, cytotrophoblast and syncytiotrophoblast, right, which are formed from this trophoblast itself. Now with this I move forward to the next one. Now once the blastocyst is formed, now this blastocyst is known as a bilaminar germ disc. Why? Because it has 2 types of germ layers. Now the embryo blast which was there, that embryo blast or the inner cell mass, it develops into 2 layers of the germ cells. Okay, and these 2 layers of germ cells is known as a bilaminar germ disc, which forms a hypoblast which is made up of cuboidal cells and an epiblast which is made up of columnar cells. Okay, hypoblast and epiblast, both of these are formed. So please remember, epiblast usually develops into most of the germ layers. Okay, please remember, most of the germ layers, they arise from this epiblast itself. Okay, Next, if I talk about the prochordal plate and the primitive streak. So please remember, there is formation of a cell mass again. The cell mass which forms at the head end or the cranial end of the embryo is known as the prochordal plate. Or we can say in other terms that the prochordal plate determines the head end of the embryo, whereas the primitive streak, it determines the tail end of the embryo. Primitive streak determines the tail end of the embryo. After this is formed, then what happens? There is rapid division of the cells, which leads to a formation of heap of cells, and this heap of cells goes between the endoderm and the ectoderm. Okay, and that leads to formation of this mesoderm, and all the three germ layers are finally formed, right? So please remember this. Now, if I talk about notochord, okay, if I talk about notochord, now you would say, what is a notochord? So this was a one-liner which has been asked. The remnant of notochord is known as. So please remember, notochord is a structure whose function is to stimulate the ectoderm to differentiate into the neural tubes. I hope all of us are quite aware that the ectoderm is the one which forms the neural tubes, okay, and the stimulus is given by the notochord. But after the function of the notochord is done, it usually obliterates and leaves behind a remnant, and this remnant of notochord is known as a nucleus pulposus. Okay, this remnant of notochord is known as the nucleus pulposus mainly. Otherwise, there is also apical ligament of Dens, but at least remember, the remnant of notochord is known as nucleus pulposus. Next, now I am talking about the derivatives of each germ cell layer. Okay, so we'll get some mnemonics also along the way, so stay connected. Now, first of all, talking about the mesoderm derivatives. Now the mesoderm, or the intraembryonic mesoderm, more clearly, it mainly differentiates into three structures. The first is the paraaxial mesoderm, second is the intermediate mesoderm, and the third is the lateral mesoderm, right? So now the paraaxial mesoderm, it generally develops into somites, and these somites will further differentiate into axial skeleton, the dermis of the skin, and the skeletal muscles. If you need a mnemonic, it is SAD. So please remember, the paraaxial mesoderm develops into somites, which further develops into SAD, where S goes for skeletal muscles, A goes for axial skeleton, that is your vertebral column, and D goes for your dermis of the skin. Next, if I talk about intermediate mesoderm, this intermediate mesoderm develops into the urogenital system. And by urogenital system here, I mean the kidneys, the testis, the mesonephric and the paramesonephric duct. Okay, I'll talk on the mesonephric and paramesonephric duct in the next session. Okay, you need to know the derivatives and the remnants of them in females as well as males. Next, if I talk about the lateral mesoderm, the lateral mesoderm further divides or further differentiates into body cavities, that is your pleural cavity, peritoneal cavity and the pericardial cavity. Whereas it also differentiates into your cardiac muscles and smooth muscles and your appendicular skeleton. Okay, appendicular skeleton means the bones of your upper limbs and the lower limbs, right? So I hope this is fine. Axial skeleton was formed by paraaxial mesoderm, appendicular skeleton is formed by lateral mesoderm. Skeletal muscles are formed by paraaxial mesoderm, cardiac and smooth muscles are formed by lateral mesoderm. Moving further, if I talk about the endoderm derivatives now, the mnemonic for endoderm derivatives is EEG. Okay, where first E goes for the epithelium of the GIT, that is gastrointestinal tract, from the oral cavity till the upper part of your anal canal, till the pectinate line. Okay, all the epithelium would be formed by endoderm. Next, epithelium of your respiratory tract, again it is formed by endoderm. And lastly, G goes for your glands. Okay, by these glands I mean the glands in the GIT, your thyroid gland and the parathyroid gland, right? All these glands are formed by the endoderm. Next, if I talk about the ectoderm now, like mesoderm, ectoderm also divides mainly into three main structures. First are your neural crest cells, second is your neuroectoderm, and third is your surface ectoderm. Okay, so if I talk about the neural crest cells, on these usually questions have been asked because here there are some exceptions. Okay, and neural crest cells, the mnemonic to be remembered is mosaic. So neural crest cells further differentiates into mosaic, where M goes for melanocytes, that is, it first differentiates into melanoblast and then into mature melanocytes. These melanocytes usually impart the pigments to the skin. Okay, it is responsible for pigmentation. You will study it in skin. Then O goes for odontoblast, which are nothing but dental cells. Then S goes for Schwann cells, and this is important to be remembered, guys. Schwann cells are derived from neural crest cells, and these Schwann cells have a peculiar function of myelination. These Schwann cells have a peculiar function of myelin sheath formation or myelination, but in the peripheral nervous system. Please remember, myelination in peripheral nervous system is the responsibility of Schwann cells, whereas myelination in the central nervous system is the responsibility of oligodendrocytes. Next, A goes for your autonomic ganglia, that is your sympathetic as well as parasympathetic ganglia. I goes for inner meninges. Inner meninges, by that we mean the pia mater and the arachnoid mater. And last, C goes for your chromaffin cells. By chromaffin cells, these usually chromaffin cells are present in the adrenal medulla. Okay, so adrenal gland usually has a adrenal cortex and adrenal medulla. In the adrenal medulla, we will find chromaffin cells which are responsible for the secretion of catecholamines, right? Adrenaline and noradrenaline. And if there is a tumor of this chromaffin cells known as chromaphinoma, it is usually seen in conditions like pheochromocytoma. Next, if I talk about neuroectoderm, guys. So about neuroectoderm, please remember, this neuroectoderm will further differentiate into your nervous systems, that is your central as well as your peripheral nervous system. That means your cranial nerves as well as spinal nerves all are derived from the neuroectoderm. And next is your retina. So retina is also derived from the neuroectoderm. If I speak further about the surface ectoderm, the third structure of ectoderm, surface ectoderm, the mnemonic to be remembered is leg. Okay, so please remember, L goes for the lens of the eyes. Okay, this is a question from ophtha also. L goes for the lens of eyes. E goes for the epidermis. Okay, E goes for your epidermis. Please remember, which is the outermost layer of the skin. Here E can also be remembered for your epithelium of your cornea as well as conjunctiva. If I talk about ophtha, please remember, here surface ectoderm also differentiates into epithelium of your cornea and conjunctiva. And lastly, G goes for your glands. So here by glands I mean the sweat glands, okay, the sebaceous glands, the salivary glands, the lacrimal glands, the mammary glands, that is your breasts, okay, and also the pituitary gland. So all types of these glands, and also the glands of your eyelids, that is those are nothing but your sweat glands in itself. Okay, so all of these glands are derived from the surface ectoderm. Right, so these are your derivatives of the three germ layers. Now this was the image which had come earlier. So this is an image of an embryo. Okay, here you can see two bulges mainly. So the first bulge that you can see over here, this is a head bulge. Okay, the first bulge that you can see over here, this is a head bulge. The next bulge that you can see over here, this is a cardiac bulge, which develops into the precordium. Right, this is a cardiac bulge. And lastly, this fold that you can see over here, it is known as a tail fold, right? So important. Whereas these two limbs that these two buds that you can see over here, these are your limb buds. Okay, the upper ones for your upper limbs and the lower ones for your lower limbs. So usually these are your limb buds. Whereas the arcs that you can see over here just beneath the head bulge, these are nothing but your pharyngeal arcs. Okay, these are nothing but your pharyngeal arcs. So important things to be remembered, guys. These pharyngeal arcs usually has three main structures. First is the pharyngeal arc. Second is the pharyngeal pouch, which is formed by the endoderm, therefore also known as endodermal pouch. Okay, this can also be remembered as the endodermal pouch. And third is the cleft, which is formed by the ectoderm, therefore also known as ectodermal clefts. So if I first discuss about the derivatives of your pharyngeal pouches, there are usually 5 pharyngeal pouches. The first pharyngeal pouch develops into the eustachian tube, the middle ear cavity, also known as your tympanic cavity, and lastly your mastoid antrum. I hope you are quite aware that antrum is the largest mastoid air cell. You will study it in ENT. Okay, mastoid antrum is the largest mastoid air cell. So first pharyngeal pouch derivative. Second pharyngeal pouch derivative is the tonsil. That is your tonsillar fossa as well as your tonsillar epithelium. Next, third pharyngeal pouch develops into thymus and the inferior parathyroid gland. So whatever I have highlighted, that means it is important or it has been asked. So please remember, the third pharyngeal pouch develops into thymus and the inferior parathyroid gland. Now there is an important disease related to this condition. So if the third pharyngeal pouch fails to differentiate, then it leads to an important syndrome known as DiGeorge syndrome. Please remember, if the third pharyngeal pouch fails to differentiate and does not form thymus and inferior parathyroid gland, that is usually seen as syndrome known as DiGeorge syndrome. So please remember, DiGeorge syndrome mein there is absence of thymus and inferior parathyroid gland, and DiGeorge syndrome, it has been asked in recent December 2021 session also. Here it is a gene deletion. So please remember, it is a 22q11 gene deletion. So please remember, on 22nd chromosome on the long arm, that is Q arm, at the 11th position, there is a gene deletion seen in DiGeorge syndrome. And this DiGeorge syndrome, it is remembered by the mnemonic catch-22, which we'll discuss further. And in this DiGeorge syndrome, there is absence of thymus and inferior parathyroid gland. Next, fourth pharyngeal pouch develops into the thyroid gland and the superior parathyroid gland. Okay, thyroid and superior parathyroid. So it is reverse. Please remember, third develops into inferior and fourth develops into superior. Whereas the fifth pharyngeal pouch, now in some books they say it is fifth, some books they say it is a part of the fourth pharyngeal pouch itself, that usually develops into the ultimobranchial body. Okay, and this ultimobranchial body leads to formation of the parafollicular cells. So this was a question which has been asked. Okay, so ultimobranchial body derivatives are, so they are parafollicular C cells. Okay, at times the question have also been asked, now recent December 2021 session again, that the parafollicular C cells are responsible for secretion of which hormone. So please remember, this parafollicular C cells, they are responsible for the secretion of calcitonin hormone. C goes for calcitonin, but usually they ask parafollicular cells only. So please remember, parafollicular cells are responsible for secretion of calcitonin, and these parafollicular cells are found in the thyroid gland. Don't confuse it with the parathyroid gland just because of the name parafollicular. Right? So parafollicular C cells responsible for secretion of calcitonin, they are derived from ultimobranchial body. But please remember, if neural crest cells is there in the option, then the best answer would be neural crest cells. Okay, so please remember, the question is parafollicular C cells are a derivative of, and you have both of these things in option, ultimobranchial body and neural crest cells. The best answer would be neural crest cells, because please remember, the pharyngeal pouches or the pharyngeal arcs are nothing but they are condensation of your neural crest cells and the mesoderm, which is formed at the floor of the mouth. Okay, so better answer would be NCC. Moving further, talking about the pharyngeal arcs now. So usually, please remember, there are total 6 pharyngeal arcs, out of which the 5th pharyngeal arc disappears. Okay, therefore there are only 5 pharyngeal arcs usually. Now each pharyngeal arc will have some skeletal component, some muscular component, some nervous component, and some arterial component. Okay, so let's discuss about these. So the first pharyngeal arc, also known as the mandibular arc, the cartilage is known as the quadrate cartilage or the Meckel's cartilage, and it derives into the first of all, the ear ossicles. I hope in the middle ear cavity you know that there are 3 ear ossicles, the malleus, incus and stapes, right? So please remember, the first two, that is your malleus and incus, are derived from the first pharyngeal arc, whereas stapes is derived from the second pharyngeal arc. Okay, so from first pharyngeal arc, malleus and incus are derived, then the anterior ligament of malleus, the spine of the sphenoid bone, the spinomandibular ligament, and the genial tubercle of mandible. At least remember the spinomandibular ligament and these 2 ear ossicles. These are most important. Next, if I talk about the muscle, so please remember, first pharyngeal arc derives into the tensor tympani muscle and the muscles of mastication. Muscles of mastication are 4 in number. I hope you know that. That is your masseter muscle, the temporalis muscle, and the medial and lateral pterygoid muscle. I'll once again repeat, the masticatory muscles are the masseter muscle, the temporalis muscle, and the medial and lateral pterygoid muscles. Whereas the mylohyoid muscle also develops from the first pharyngeal arc and it forms the floor of the mouth, mylohyoid muscle. Then the anterior belly of digastric muscle develops from first arc, whereas the posterior belly of digastric muscle develops from the second arc. Easy to remember. And last muscle that develops is tensor villi palatini muscle. Next, if I talk about the nerve, so please remember, first pharyngeal arc develops into the third division of the trigeminal nerve known as the mandibular division. Now I hope you know that the trigeminal nerve, which is the 5th cranial nerve, has 3 main divisions. First is the ophthalmic division, second is the maxillary division, third is the mandibular division. So the mandibular division is derived from the first pharyngeal arc. And lastly, it develops into a artery. So please remember, usually it develops into the first aortic arc artery. right? Or you can say it as the maxillary artery. So please remember, it is the maxillary artery, not the mandibular artery. Okay, yeah, that now. Next, if I talk about the second pharyngeal arc, it develops in it is also known as the hyoid arc, and it develops into a cartilage known as the Ricard's cartilage. Here it develops further into the stapes bone, which is the smallest bone inside the body, and the muscle related to it is the smallest muscle inside the body. So smallest bone inside body is stapes, and smallest muscle inside body is stapedius. Then it also develops into the styloid process of the temporal bone, the stylohyoid ligament, and the lesser horn, and the upper part of the body of the hyoid bone. Whereas in muscles, if I talk about, it develops into your stylohyoid muscle, your facial muscles like buccinator, platysma, and the posterior belly of digastric. Whereas it develops into your seventh cranial nerve, also known as your facial nerve. And lastly, it develops into the stapedial artery, right? Next one, the third pharyngeal arc now. The third pharyngeal arc usually develops into the greater horn of the hyoid bone and the inferior surface of the body of your hyoid bone. And it also develops into the stylopharyngeus muscle. Baki sub S, please remember, most of the S are seen in the second pharyngeal arc except one S, that is your stylopharyngeus muscle. Whereas the nerve which is derived from the third pharyngeal arc is known as the glossopharyngeal nerve or your ninth cranial nerve. And lastly, it derives into the common carotid artery and the internal carotid artery. Then if I talk about the fourth pharyngeal arc, it usually develops into all the laryngeal cartilages except one. So please remember, fourth pharyngeal arc develops into all laryngeal cartilages. I hope you are quite aware. Question have been asked on cartilages of larynx also. There are 6 cartilages of larynx, 3 paired and 3 unpaired. So all the 5 cartilages like thyroid, corniculate, cuneiform, epiglottis and cricoid are developed from the fourth pharyngeal arc, but only one, that is arytenoid cartilages, they are developed from the 6th pharyngeal arc. Next one, if I talk about, all the pharyngeal muscles are developed from the fourth pharyngeal arc except one, that is stylopharyngeus, right? We discussed it already. And the extrinsic muscles of larynx from fourth pharyngeal arc, whereas intrinsic muscles of larynx from the sixth pharyngeal arc. Extrinsic from fourth, intrinsic from sixth. Next, if I talk about the nerve now, okay, the 10th cranial nerve, also known as the vagus nerve, it has its pharyngeal branch which develops from the fourth pharyngeal arc, and this pharyngeal branch, along with this also the superior laryngeal nerve develops from the fourth pharyngeal arc. So please remember, superior laryngeal nerve develops from fourth pharyngeal arc, whereas the recurrent laryngeal nerve develops from the sixth pharyngeal arc. Next, if I talk about the fourth pharyngeal arc, please remember it develops into the arc of aorta mainly. Okay, it develops into the arc of aorta and the right subclavian artery. Whereas the sixth pharyngeal arc, it usually develops into the pulmonary trunk. Sixth pharyngeal arc develops into the pulmonary trunk, right? Then next, if I talk about the pharyngeal cleft. So please remember this table, at least if you could not remember all of that, at least the ones that I've marked as important, that should be remembered. Okay, so please remember, I don't think so there's issue in remembering the nerves at least. First arc into mandibular nerve, second arc into facial nerve, third arc into glossopharyngeal nerve, fourth arc into SLN superior laryngeal nerve, and sixth arc into recurrent laryngeal nerve. Right, they can ask you about stapedial artery. Second, please remember stapes all is second. Stapes bone, stapedius muscle, stapedial artery is the second pharyngeal arc. Mandibular, anything related to mandible is usually the first pharyngeal arc, right? At least these you should remember, I would recommend you. Next, if I talk about the third part of the pharyngeal arcs, that is your pharyngeal clefts, which are derived from the ectoderm and therefore also known as the ectodermal clefts. So usually they are 4 in number, 1, 2, 3, 4. But only the first pharyngeal cleft, it gives its derivatives, and usually the first pharyngeal cleft develops into the pinna and the external auditory canal. Okay, the pinna and the external auditory canal are formed out of the first pharyngeal cleft, whereas second, third and fourth pharyngeal cleft, the second, third and fourth pharyngeal cleft, they usually obliterate and they form the cervical sinus. Okay, so this cervical sinus usually obliterates after birth. But what if this cervical sinus persists after birth? So please remember, if this cervical sinus persists after birth, then there can be accumulation of some fluid inside, and that can lead to a neck swelling, right? And this neck swelling, which is seen due to persistence of the cervical sinus, it is usually an anterolateral neck swelling. Okay, anterolateral neck swelling on the anterior border of the sternocleidomastoid muscle, and this condition is known as the branchial cyst. So please remember, this condition is known as the branchial cyst. So this question has been asked. Branchial cyst is due to so please remember, branchial cyst is due to persistence of the cervical sinus, or we can say due to persistence of the pharyngeal clefts. Okay, so this is an important question from surgery. Nowadays, please remember guys, in preclinical or paraclinical subjects, questions have been asked to you in an applied fashion. Okay, so applied aspects are usually asked commonly rather than just factual questions. Yes, definitely there are a number of factual or fact-based questions as well, but mostly their deviation is slightly towards the applied aspects. Okay, so with this, I hope I end the session for today, and in the next session we'll be discussing about important topics like the mesonephric, paramesonephric duct, the cardiovascular system, embryology somewhat. Okay, so other topics like these miscellaneous ones regarding embryology will be discussed in another session. Till then, thank you so much, goodbye, good luck, and let's meet you in another session.